Inbreeding is a type of non-random mating that occurs when related individuals mate more often than would occur by chance. This increases homozygosity, but does not change allele frequency. Let's take a look at how allele frequency is not affected, even though phenotypic and genotypic frequencies will be affected. When dealing with a single gene, two different alleles are possible, generally denoted by a large letter and a small letter. Within a population, there are three different genotypes that are possible. Big A dominant, big A heterozygous, which will still show the dominant phenotype, and little a homozygous recessive, which will show the recessive phenotype. When inbreeding occurs, it's as if each individual was mated with itself. So homozygous dominant will mate with another homozygous dominant, heterozygous will mate with heterozygous, and homozygous recessive will mate with homozygous recessive. This is the parental generation. We can use a simple Punnett square to see the results of each mating. With the homozygous dominant, all homozygous dominant offspring resulted. With the heterozygous mating, there was one homozygous dominant, two heterozygous individuals, and one homozygous recessive. And with the homozygous recessive individual, all homozygous recessive progeny resulted. If we count the number of alleles in each set, we can see that there are 4 big A's in the first set, 2 in the second set, and then 2 small a's in the second set, and 4 in the third set, making it 6 big A's to 6 little a's, or a 1-to-1 ratio. This would give 50% of the alleles to be big A and 50% of the alleles to be little a. Taking a closer look at the Punnett squares will help us understand how the genotypic frequencies are changing, but the allele frequencies are not changing. Each homozygous individual that was selfed will clearly have all of the same progeny each time. The heterozygous individual, however, will have half of its progeny like itself as a heterozygote, and 1/4 of them will be homozygous dominant and 1/4 will be homozygous recessive. This chart will help us visualize this a little better. In the parental generation, or generation 0, we can see that the homozygous individuals produced offspring just like themselves. The heterozygous produced half like itself and 1/4 like a homozygous dominant and 1/4 like a homozygous recessive. If this continues each time, the heterozygous population will be cut in half, and a 1/4 more will be given to each of the homozygous recessive and homozygous dominant populations. This will result in separation of the genotypes and a pure breeding line of homozygous dominant and a pure breeding line of homozygous recessive, with a decrease in the number of heterozygotes in the entire population. However, each of the allele frequencies will remain constant. As we can see here, that P and Q will have stayed at 0.5 throughout the entire time, even though the number of heterozygotes has decreased. This is because at each separation, or each generation, there are an even number of alleles, big A alleles going to the big A side and little a alleles going to the little a side, so no side is ever dominating the other. I hope you learned a little more about inbreeding and how it can increase homozygosity of a population, but does not change the allele frequency over the entire population. So thank you, genetics, for teaching us so much about life.